Hi, I'm Ronen Zaidel-Bar. Welcome to my lab. And now, I'd like to pass you over to Tim, who will explain to you more about what we do in the lab. Our lab, which is a part of the Department of Cell and Developmental Biology at Tel Aviv University, works at the intersection of developmental biology and cell mechanics. We study how cells and tissues get into shape during development. More specifically, we focus on the cytoskeleton, which is made of filaments such as actin and microtubules, as well as motor proteins, adhesion sites, and the extracellular matrix. What we are figuring out is how these cytoskeletal elements work together at the right time and place so that cells can divide, change shape, generate forces, and move around as necessary for normal animal development and physiological function. Failure of these cytoskeletal machineries to work properly results in a variety of conditions, including cardiovascular disease, neurodegeneration, pulmonary fibrosis, and skin blistering. To study all this, we use an awesome little nematode called C. elegans. C. elegans, also known as the worm, is an ideal model organism thanks to its small size, short life cycle, wealth of genetic tools, and very importantly, its transparency, which facilitates microscopic imaging. With only 1,000 cells in the adult hermaphrodite, C. elegans still has an elaborate anatomy, including skin, muscle, digestive, nervous, and reproductive systems. Thus, C. elegans is at a sweet spot between complexity and simplicity. It is complicated enough to be relevant for human biology, and yet simple enough for us to understand it in our lifetimes. Importantly, 80% of human protein-coding genes have orthologs in C. elegans, and the majority of human disease genes are present in worms, making C. elegans an excellent model to understand the molecular basis of human disease. What does this actually look like in real life? Designing a new CRISPR-Cas9 project to introduce a point mutation or a fluorescent protein into a specific gene is done with a special website we created called Worm Coolkit. Handling of the worms takes place on a stereo microscope. We grow worms on agar plates and move them from plate to plate with a platinum wire pick. One way we learn about the function of a protein is by observing what happens when we knock it down using a technique called RNAi. Another way to learn about protein function is by tagging it with a fluorescent protein and then observing its localization and dynamics under the microscope. Making transgenic worms involves injecting DNA and Cas9 protein into their germline, a task that requires a very thin needle and great dexterity. To image live worms, we have our own spinning disc confocal microscope. With it, we can make movies following the dynamics of proteins within the body of the developing embryo or worm. Our lab is located in the Faculty of Medical and Health Sciences building at the north end of Tel Aviv University. Our lab members are postdocs, PhD and master students, and undergraduate students that come from all around the globe and are the most creative and hardworking students in the department, if I may say so myself. We're a medium-sized lab, so everyone is close with each other, and we even hang out together outside of the lab. Tel Aviv University is a powerhouse for research in the biological sciences, and its students are boundlessly curious and maintain a good life-work balance. And Tel Aviv is an incredible city. While it is the technological and financial hub of Israel, it also has superb beaches, restaurants, arts and museums, nightlife, and public transportation. It is truly a bustling city, built for young people on the go. Thank you, Tim, for those excellent explanations. Please, if you have any questions or would like to join the lab, don't hesitate to reach out.